And welcome to my YouTube channel Biology Articles and MCQs. Guys, our today's topic of discussion is enzyme inhibitors, and we will also discuss its type. So let's start. First of all, that what are the inhibitors? Guys, inhibitors are any chemical substance that can react with the enzyme, but in place of substrate. And after the reaction with the enzyme, it will not transformed into the products. As you know that the enzyme when react with the substrate, it will form a complex that is called as enzyme substrate complex. And after the formation of this complex, the substrates are then transformed into the small parts that are called as products. But in case of inhibitors, when the inhibitor will attach to the enzyme in place of the substrate, it will not be transformed into the product. Means in case of substrate, the inhibitor will not transformed into the product and the products will not be formed. So as the products will not be formed, so the active site of the enzyme, active site, you know that it is a site of the enzyme at which the substrate attach. So the active site of the enzyme will block, and this blockage can be temporary or can be permanent. So guys, we can say that the inhibitors are the agents that can slow or interfere with the enzyme action because it is suppressing or reducing the activity of enzyme. As the activity of enzyme is enhanced by the presence of the substrate when it will be transformed into the products, and as the inhibitors are not transforming into the products, so you can say that it is interfering with the enzyme action. And if we talk about that what are some examples of the inhibitor, then the inhibitors can be poisons, or it can be antimetabolites or some cyanides, and even some drugs and antibodies also act as can act as inhibitor. So moving towards the types of inhibitor, there are main two types of inhibitor: irreversible inhibitor and reversible inhibitor. Let us discuss the first type, that is irreversible inhibitor. Guys, as its name shows, when this type of inhibitor will be attached to the active site of the enzyme, that action cannot be reversed. Irreversible shows that that action will not be reversed. Mean as it will attach to the active site, it will attach permanently, so that it will not leave the active site, and then later the substrate also cannot attach. Here you can see that there is a inhibitor shows, and this inhibitor has the structure that is related to the substrate. So when it will attach to the enzyme, here it is an enzyme, and this is the inhibitor. It will attach this part, this triangular part to another portion, and this part to the active site. And due to its structural similarity, it is occupying the active site by forming the covalent bonds. And you know that the covalent bonds are very strong bonds, so this active site will be physically blocked, and this action cannot be reversed. Means this inhibitor will not leave the active site now. So guys, these inhibitors may react with the sulfhydryl groups of the enzyme also. Here is a sulfhydryl group as such that is shown, that is attaching to this enzyme, and this is one inhibitor shown here. And when this inhibitor will react with the sulfhydryl group, here you can see that this inhibitor has attached to this sulfhydryl group, it will physically block the active site, and then then precipitate the enzyme. Mean it will deposit it in the solid form. So the irreversible inhibitors, when attached to the active site, they will form the covalent bond that are very strong bonds, and to break these bonds is not possible. So the second type of inhibitors are the reversible inhibitors. Similarly, as its name shows, that when these type of inhibitors will attach to the active site of the enzyme, these action can be reversed easily. Means these inhibitor can be neutralized, or their effect can be neutralized completely or partly by the increase in the concentration of the substrate. Means when we will increase the concentration of the substrate, that inhibitor will leave the active site and that substrate will attach to the active site of the enzyme. So guys, the reversible inhibitors have further two types: competitive inhibitors, non-competitive inhibitors, and uncompetitive inhibitors. The competitive inhibitors are the types that have structural similarity with the substrate, and due to the structural similarity with the substrate, they can be easily selected by the binding site. Means it will attach to the binding site of active site. Guys, you know that there are two further sites of the active site. One is binding site and the other is catalytic site. The binding site will help in the binding of the substrate, and the catalytic site will help the catalysis. Means it will change the substrate into the products. So this inhibitor, as it is structurally similar to the substrate, it will attach to the binding site, but it is not able to catalyze the reaction. Means it will not transformed into the products. Here you can see that there is a inhibitor and it is now attached to this active site. Let us discuss it with an example. Here you can see that there is a succinate, and this succinate is a substrate, main substrate, and succinate is a 4-carbon compound, and here is the succinate and enzyme substrate enzyme complex is formed, and as a reaction of this complex, the product that is formed is fumarate enzyme. But you can see that here is a malonate, that is an inhibitor, and you can see that also that it is structurally similar to the succinate. Succinate is a 4-carbon compound, and the difference in the succinate and malonate is only the difference of 1 carbon number. Means the malonate is a 3-carbon compound. The functional groups of succinate and malonate are completely same. So this malonate inhibitor can be easily selected by the binding site, but in this case no product will be formed. The second type of the reversible inhibitor is non-competitive inhibitor. Guys, we see that in the competitive inhibitor, the inhibitor attached to the active site. But in case of a non-competitive inhibitor, the inhibitor will not attach to the active site. It will attach to an other point other than the active site, and this type of site is called as allosteric site. As you can see there, if the normal substrate is present, it is now attaching to active site here. But if the inhibitor is present, then it is attaching to this site, and this is not the active site because it is site another than the active site. So it is called as allosteric site. And it will alter, or it will change the structure of the enzyme in such a way that even if the genuine substrate binds the active site, then catalysis fail to take place. Moving towards its example, here you can see that there is an inhibitor, that is Hg+2 ion, mercury ion, and it is now attaching to this site, and this site you can see that this is not the active site. Here is the active site shown, and here is another site with 2 sulfhydryl groups here, and when this mercury ion will attach with these sulfhydryl groups, here is such structure will be formed, and this structure is forming in the allosteric site, that is site other than the active site, and as a result of this the structure of the active site is also changed. And when the availability of the substrate even if increases, genuine substrate, and it will and even if it bind to the active site, then catalysis will not take place, mean it will not change into the products. So this is called as non-competitive inhibitor. The third type of these inhibitor, the reversible inhibitors, is the uncompetitive inhibitor. And guys, the uncompetitive inhibitor is don't attach to the active site, or it will not attach to the enzyme directly. It will attach to the enzyme substrate complex. Means, first of all, the substrate will attach to the active site of enzyme, and when the enzyme substrate complex will form, this inhibitor will attach to that enzyme substrate complex, and so this type of inhibitor is called as uncompetitive inhibitor. So this is all the discussion about the reversible and irreversible inhibitors. I hope you understand all the things about it, and don't forget to share this video with your friends. Thank you so much.